Thank you very much for the invitation. I am sorry in advance because I will not be able to talk about the TADs today. So the focus of this talk is chromatin architecture in sperm. And so let me see. Yeah. So here's the global picture you all have in mind about what the chromatin looks like in the nucleus, right? And so you move from nucleosomes to territories to compartments and to the noodle soup. And the noodle soup that I know all of you guys are very excited to look at this this checkerboard, and it's really so I have kind of a different perspective on this because sperm cells are so widely differentiated in nature that this architecture just pales in comparison, right? So here you just have a few examples of what sperm looks like in different species. And you can get these needle kind of architectures. You get these round cells, but you can also get this kind of like bottle opener, right? So it's a structure function that is pretty clear here what the sperm is doing, right? And so what's really interesting about this is that the genome needs to fit into this in an architecture that is both morphologically very specific. It's also very compact. Because the sperm has to be motile, it has to go into the egg. And the main mission of the sperm is, right, to transmit the intact DNA from the father to the egg, right? And so in Benjamin's lab, we asked the question of how this works, right? And so part of the answer, maybe a big part of the answer, comes from the fact that chromatin in spermiogenesis becomes very, very deeply remodeled in a process where in many animal species, you will almost completely lose the histone proteins that make nucleosomes in somatic cells, and you will replace them by specific proteins, in particular the protamine family. So you can see here, for example, in Drosophila, how the histones will first become hyperacetylated. Remember this detail, it will become important. And then these histones are actually removed from DNA and replaced first by transition proteins, which are highly basic proteins interacting with DNA, and then by protamines. So the protamine family of protein is actually highly evolving, but it has several key features that are conserved. So one of them is that there tend to be short proteins that are highly basic, right? So they are very rich in basic residues, arginine and lysine. And they also have a pretty high amount of cysteines in general, although this is not a completely absolute. So if you just take protamines and DNA and you put them into a tube, what will become is DNA will form a toroid architecture, which is kind of a property that is expected if a multivalent cation becomes in contact with DNA. But what is interesting is that cysteine residues between protamines can actually make covalent disulfide bonds. And so the protamines start to cement on themselves and on each other. And so people have come up with models by which there is a very frozen chromatin architecture that is enabled by the fact that protamines kind of stick to one another once the architecture of the sperm is done. So, like I said, in the lab, we are interested in trying to understand how this works. And so crickets turn out to be a really nice model for this. And so here you can see on the left, here the actual crickets, it's almost the right size. It's actually the size of a thumb in my screen. And so here are the male crickets. And we like crickets as a models for spermiogenesis because the testicles in crickets are huge. They have like a huge testicles. So it's almost 15 or 20% of the adult body mass in some instances. And so in each of these testicles, you have hundreds of these follicles that you can see here. So this is just DNA staining of a testicular follicle. And here you can see the progression between early cells, spermatocytes, moving on to differentiation. So they will first become haploid round spermatid after meiosis. So round spermatids are small and pretty compact. And then in crickets, and this is also pretty common in insects, the sperm cell will acquire a very specific needle-like morphology, so that it will become to elongate more and more. So it becomes first this kind of a canoe shape, and then it becomes this needle that is actually 40 times longer than it is thicker. And again, what's really interesting is that the DNA needs to fit into this very particular nuclear architecture, right? So in crickets, there is evidence that there is this histone to sperm-specific protamine transition. One of the reasons why we think so is that we completely lose histone signal as we move on during spermiogenesis. So you can see here that the histones that mark very well the early elongating spermatids will no longer mark at all the nuclei in later cells. And what also really got our attention in crickets was this study from the 1980s. So in the 80s, people did a lot of electron microscopy, and they look a lot into sperm of many insects. And in this particular brand of crickets, Gryllus bimaculatus, we see this really extraterrestrial architecture where the chromatin seems to be doing these fibers that really are elongated along the axis of the nucleus, and they go around the nucleus like they were bundles of chromatin fibers. Really very strange, right? And so our first mission was to try to see how this actually happens over time in spermiogenesis. So with the help of Dominique Bass in Lyon, we did electron microscopy to look at the different stages in spermiogenesis. So here you have the early cells, which correspond to the DNA noodle soup, that like the somatic organization with nucleosomes. That kind of looks like this. It's kind of messy. You don't see much features apart from this mass of heterochromatin at the border. But then when we look at elongating spermatids, you can see that very quickly we acquire this ordered organization, this orientation of chromatin fibers that really stretch along the nuclear axis. And if we look at later cells, so this is kind of the canoe stage I was showing you before, it's pretty elongated. We can see that the fibers are really stretching all along the nucleus. This is a sagittal section of that nucleus. If we do a zoom into these fibers right here, you can see that these chromatin fibers are actually about 30 nanometers thick, and they are hollow. So we figure that the DNA is actually twisting around inside of these fibers. If we look at transversal sections of this sperm, you can actually count all the little fibers going straight from one end to the other. There's more or less 200 of these fibers at this particular stage. If we look at the acrosome region, so this is the head of the sperm, we can recapitulate the observations that were done in previous studies, where the fibers get to the head and then they kind of turn and go back. And then at the other end, at the basal end, which is where the flagellum of the sperm is anchored. So the sperm is motile because it has this long microtubule that is like the sperm tail. We see the fibers going down onto the flagellum and anchoring at the bases, right? And so you can actually see that there is, in this picture, that there is kind of an overlap between the flagellum and the chromatin fibers at the section of the nucleus, right? So this is a very special, very unique architecture in these cells. And the electron microscopy can only go so far, because at some point the compaction of chromatin inside the nucleus becomes so dense that there's just too much matter. And even in electron microscopy, we don't see any detail anymore. We just see a kind of a black mass of material. And so to overcome this, we implemented a second approach, which is a storm microscopy. So this was thanks in particular to a collaboration with Jean-Philippe Clément in Grenoble. So just very briefly to recapitulate the principle of storm, many of you probably know this, but the principle is that if you have many points of light that are emitting light at the same time, light is limited by a physical property of resolution, the diffraction limit. And so you will see all these dots massing up into a blob of light. But so what storm microscopy does is that you can shut all of the lights down in all of these luminescent points, and then you can turn them on one by one. And so every point is still limited in the resolution you can get by the diffraction limit. But now, because it is very round and very clear, you can mathematically calculate the middle coordinate of this spot of light. And so you can more precisely map the position of molecules. And so if you take a video of molecules that are blinking, you can reconstitute a super resolved image. So, so the Eiffel Tower, right? So if you have lights in the Eiffel Tower and you look at them one by one here, and so you reconstruct an image there, you can see how you get a more a more precise, right, picture of where every spot of light is. So we use this method to map the organization of the genome inside the sperm cells. And we used a DNA labeling probe that was developed by our collaborators, Grasvidinias Lukinavicius, so which is a hydroxymethyl siliconized rhodamine coupled to Huxt. So it's a dye that can blink and label DNA. So we do this kind of a super resolved analysis of DNA organization in elongated nuclei, where, you know, as I showed you before, electron microscopy would not allow us to see much. And here we can actually see how the chromatin fibers are actually starting to make these twists, right? That I hope you can see that clearly here. We see large scale twisting of the chromatin fibers, bending around the nuclear axis. So this is a relatively late stage in spermiogenesis. This is an even later stage in spermiogenesis. You can see the twisting going around. So to recapitulate this description of what's happening in the nucleus. So, so this is actually something my daughter did. So it's pretty visual, I figure. This was a proud daddy moment. So you get a noodle soup first, and then the noodle soup will organize into this bundled coil of fibers, and then it will twist around. And so mechanistically, to understand how this works, to me, this is like literally magic, right? And so to try to get kind of an insight into how this works, we turned to people who know physics. So this is Daniel and Maxime at our lab at the LBMC, who implemented a polymer model to try to describe the movements of chromatin in this developing cell. And so the polymer model is, they tell me, relatively simple, which is it's just a string of beads, which are more or less 30 nanometers wide. And they have a certain propensity to touch each other, and they have a certain, so they confer to the chromatin a certain rigidity. And this polymer evolves within a discrete membrane. So it's just a membrane that is fixed, and then we can play with the membrane parameters to kind of allow or not the extension and the deformation of the nucleus. So after a few trials that I will not really describe, what Maxime says was that there is one thing that could explain this coiling of chromatin, this nematic ordering of fibers, which is if the fibers suddenly became rigid. And so rigidification is actually compatible with what we expect if we replace the histones by the protamines, which are more basic, right? And so he simulated what would happen with the polymer if all of a sudden all the chromatin fibers became rigid. So this is this simulation that you can see here, starting, as you saw, with the noodle soup. And so as the simulation progresses, you can see that there is indeed some ordering of the fibers that is forming, that is kind of pushing the external fibers into being relatively parallel to one another. So here's just a few snapshots of these simulations. And so this simulation actually kind of recapitulates some interesting features that we see in vivo. So here's again the noodle soup, and at this stage, which is more ordered, this would correspond to the more differentiated round spermatid, which is also more compact. You can see that both in the simulation and in vivo, we see this higher density of material at the periphery of the nucleus. So this simulation becomes kind of compatible with what we see in vivo. But then if we take this nucleus, and so you already see it's kind of messy, right? And if we kind of try to stretch the nucleus to make it elongated, like in the cricket, we find an architecture that is really a bit too messy compared to what we actually see in vivo. So in vivo, in early elongating and even really very early and early elongating cells, we already see an ordering that is pretty sharp that we do not recapitulate in this kind of simulation. So we knew that the simulation was more or less incomplete. And so we were thinking about how to improve the model, and we made an observation in vivo, which is about histone acetylation pattern. So you remember I told you that before you can remove the histones and replace them by protamines, histones need to become acetylated. And in flies and in mice, this happens kind of at the scale of the whole nucleus. But we discovered that in crickets, this happens in a very spatially defined pattern. And so in early cells, you will see spots of acetylation coming in, but then this will group at the middle of the nucleus, like you see here. And then you will actually see kind of a wave of acetylation going on two poles of the cells, right? So it's very clear in these kind of examples here. To later become a little bit more fuzzy when the nucleus becomes elongated. It's interesting to note that when the acetylation is doing this kind of wave, DNA is actually localizing more at the periphery of the nucleus, right? Not exactly where you see the acetylation. So this gave the Maxime and Daniel the idea that the rigidification could be the explanation, but it could be more subtle than that. It could be nucleated at some specific points, representing the acetylation. So they revised the model so that the rigidification was nucleated. What we mean by that is that at some point, an arbitrary portion of all the chromatin fibers will become more rigid. And then, so this is represented by they becoming green in the model. And they become darker green, which is they become more and more rigid. And once they become blue, this means that they are fully rigid, between brackets. And so they can infect by rigidification the two neighboring fibers, right? So this kind of spreading of histone modifications is something that is very well described for many histone modifications, including acetylation. So it's reasonable to think that something like this could be going on. And so Maxime started a second set of of simulations. So here in light green, you can see the fibers that are starting to get rigid. And then in blue, you can see the fibers that have become fully rigid. And what you can see is that these fibers that are rigidified, they are localizing to the periphery, and they are starting to become parallel to each other. So this effect is actually much more ordered than what we saw before when we first thought that all of the fibers change their property at the same time. And so to recapitulate, let me see, right. So to recapitulate what happens with this model, here on the top panel, you see only part of the fibers. Here you see all of the fibers. But basically, what we see is that there is a very clear pattern of where the rigidifying chromatin should be localized in the nucleus with a spool of chromatin really forming around the nucleus, but with a certain polarity, right? Because if we take this kind of nucleus, we see that there are very parallel fibers on one orientation, and then on the other orientation, in the middle of the nucleus, it's really like there was a hollow spool. There is more disordered chromatin. And so this proceeds until everything becomes rigid and everything becomes much more well-oriented than in our previous simulations. And here I want to point out that what we think is really a striking similarity between intermediate stages in our modeling, where we really see this kind of poles of acetylation going on, and what we saw in vivo right. So we really do believe that there is a nucleated rigidification of chromatin, whether it is actually acetylation that is doing this, or it is the transition between histones and transition proteins and protamines. We haven't figured it out, but it's reasonable to assume that this is likely to be the mechanism. So a second question is, so here what you can see is that in this cyst of cells, so all the cells are very coordinated, right, in this tissue. You can see that there are different orientations, right? So here the pole of the cell is left to right, and here it's north to south, right? And so one of our questions was whether chromatin could impose the orientation to the cell, or actually it was the other way around. And part of the answer came from other observations in electron microscopy, where, for example, here, what we saw in this early cyst, where the spermatids are round, so they are early spermatids, you can see in this cell, for example, the baby acrosome, right? So this is the baby head of the sperm that is just starting to grow. And at the opposite side, you can see the baby tail of the sperm, the baby flagellum. And so in a cell that is already oriented in the posterior anterior axis, you can see that the chromatin itself is actually not oriented, right? So the chromatin here is seems to be really symmetrically, spherically symmetrical. And so the idea then was that the cell became oriented. And so there had to be a mechanism by which the chromatin followed the cell orientation. So the spool of chromatin became oriented in the right direction. And so Maxime did other simulations where he forced the elongation of this cell. And so you can see in this, so I don't actually show you the video because it goes so fast, but in this simulation, even when the chromatin orientation starts really at the worst possible orientation, which is perpendicular to the elongation axis, the chromatin will very, very quickly become oriented in the right direction and become this filament of coil. So here is a configuration that is much more ordered than what we saw in the first simulations. And you can see here, if we do a transversal cut, that we see kind of the remaining of the spool orientation we saw before, with the chromatin fibers being parallel in one direction and perpendicular in the other. And so this is something that we also see in electron microscopy, which is pretty reassuring. Right, so remember that before I showed you that the chromatin did yet another trick, which was to twist along around its axis, right? And so we actually never really figured out what could be the mechanism to impose on this twisting. But what Maxime could do is to force this twisting by forcing geometrical constraints into the polymer and the membrane, right? And so if he did that, the polymer became twisted, like we knew was happening in vivo. And if now he released these twisting constraints, something that was pretty surprising is that the first reflex of the polymer is to compact even further, right? So this configuration is kind of favoring the fact that once you release the forces, the polymer will tend to collapse on itself on this twisted configuration. And you can see here that this could actually correspond to successive stages in spermatogenesis, where we see that when the fibers are twisted, we see these fibers kind of laying down perpendicular to the axis of the nucleus. This is a transversal section of the elongated nucleus. But when the fibers become really compact, this is when we actually cannot see anything anymore, because the matter is just too dense inside this nuclei, right? So like I said, we don't really know what could be causing this torque, this twisting, but we do have an idea, which is that maybe the flagellum could be applying this force. This is purely speculative at this point, but what we do know is that there is chromatin fibers that actually become anchored to a cytoskeleton that is connecting the tubular flagellum to the chromatin. And at intermediate sections of the cell, so you can see here, if we do a section in the body of the nucleus, we can see actually that the flagellum is really penetrating inside of the nucleus. So there is a point where it's completely surrounded by chromatin. So we have no proof of this, but it's reasonable for us to think that because the flagellum is twisting, its torque could be applying a force to the chromatin fiber that makes it twist on itself and ultimately brings just the final punch to the ultra compaction of the sperm. So once this happens, right, like I said, there is the idea that this architecture needs to be maintained, and one of the ideas was that it could be maintained in the needle shape by the force of protamine disulfide bonding. So this is work by Bea in the lab, who what she did was to actually probe what would happen if you break the disulfide bonding with protamines And you can do this chemically by exposing the cells to DTT, which is a very broad-acting reducing agent. It does other things, but in these cells, it is expected to do a lot of mess inside chromatin. And so here it's a lifetime video. It's a real-time video, sorry, that you can actually see that once you add the reducing agent, the chromatin breaks down really, really quickly, right? So this process is actually, this process of decompaction that you saw is actually happening in a matter of seconds. And so in the simulations, the polymer simulations, if you release the applied forces to this chromatin configuration, you will see this kind of intermediate stages, and then go back to this pool, which is kind of reminiscent of the intermediate stages that we do see. So here you can see the twist, untwisting, right? And so it ends up being fully decompacted. So I guess that all of this talk is kind of an anecdote, right? But I really love this work and in collaboration with the physicists, because it really puts into perspective what's the way we think about genome organization in a cell. So in this particular species, the sperm chromatin organizes in a very weird way for thick fibers that can coil and elongate along the nucleus axis. And what is really striking is that most of this organization can be explained by a pretty simple set of biophysical transitions. Namely, you just need to rigidify chromatin in a nucleated fashion. And then how nature really contributes to itself when it achieves this twisted configuration that apparently can achieve a maximum level of compaction and solidity for the DNA to safely travel between the testicle and the egg. Right? So with that, I want to acknowledge again all the cricket team that took care of all the work, especially of course people at the ENS, so Benjamin, Beatrice, and our physicist buddies Daniel and Maxime, who will, I hope, answer all the questions you may have about physics, because I will be pretty much useless on that. And also to say that, yeah, I'm starting a lab at Grenoble. There are postdoc opportunities available, so please write if the keywords chromatin and crickets sound interesting to you. Thank you very much.
So Frederic is asking for rigidification. Would the acetylation of histone H4 decondense chromatin and facilitate the protamine incorporation, meaning that acetylation will the gateway to allow DNA to be available and being rigidified by protamines?
Yeah, so thank you, Fred, for that question. Yeah, I think that we don't have an answer for you, but it could actually be both in theory. So, you know, H4 acetylation, or I mean, acetylation of histones is a chemical modification that is expected to open chromatin because it kind of pushes away the nucleosomes that are surrounding. So in that sense, in a very local, very nucleosome scale, it facilitates DNA accessibility, and right. But if you look at this in a bit more macro view, there are work that suggests that actually acetylation could instead rigidify chromatin, simply because it actually prevents that chromatin bends on itself, right? Because acetylation kind of pushes itself away. So I think we're kind of at the limit of that. We don't have a clear answer for that. But just to say that histone acetylation could be a factor of rigidity by itself, although this is kind of ongoing in the field. But the second thing is that, of course, we also see that histone acetylation is the first step before you replace the histones by transition proteins, which themselves should confer more rigidity to the fiber, because these are very basic proteins that, other than that, don't have many contacts, right? So in order to answer this question, we need to actually identify the transition proteins and the protamines and tag them and see where they localize in the nucleus. So of course, this is things that we're trying to do, but yeah, ongoing.
Okay, so thank you, Guillermo. This is absolutely fascinating and beautiful work that you've presented. My interest as a cytogeneticist, who's also really interested in sperm structure and function, is about the more general karyotype and chromosome level dynamics. So how many chromosomes do these crickets have? How might variation in the size and number of chromosomes affect this spooling process?
Yeah, so crickets have 46 chromosomes. The genome is more or less 1.7 gigabases. The crickets are an X0 species. So we actually see what we think is X chromosome. Meiotic sex chromosome inactivation. This is the mass of heterochromatin I was showing you before. So this is to answer more of a general question about the karyotyping, but to answer the specific question, this is really a big mystery for us, right? Because it would be super interesting if the chromosomes are arranged within this configuration in a stereotypical manner, right?
That's kind of the second half of my question, which is, yeah, in the vertebrates, we know that there's some conservation of chromosome territories in sperm. It looks like this spooling process kind of smears or homogenizes the chromatin fibers throughout the nucleus.
Absolutely, yeah. So just to rephrase it, so in mouse, for instance, where this is very well studied, you can see that the compaction of chromatin in the sperm is much more homogeneous, so it becomes denser and denser and denser. But people have described that there was this maybe small scale toroids and small scale rods of chromatin, but you don't really see this very large scale type of reorganization in mice. And in mice, when you do a high C experiment on sperm, you see that it's more or less the same than any given somatic cell. I cannot imagine that this should be the case for crickets. It would be really beyond understanding. Unfortunately, the genomics of crickets is something that is just at its very early stages. The genome was published this year. So now we're starting to do epigenomics. We plan to do high C and stuff, and hopefully we can have more precise answers about that. But yeah, the chromosome territorialization within the sperm should be a fascinating question in this particular
Absolutely.
case.
Oh, good luck with it. It sounds brilliant.
Thanks.
Hello, I'm absolutely excited, and as a morphologist, I adore what you are showing. But I must say, your daughter was a collaborator, but my son was a collaborator at Sperm, human sperm and fertility questions 20 years ago. And we began with that we studied spermatogenesis in a rat. And what I want to say that elongated spermatids, what we found, when the histones are replaced by protamines, there are introduced DNA strand breaks. It is known. And these strand breaks are transiting from one pole to the other. We've shown it by Acrosome orange staining. So, and this allows replacement. So, in addition to acetylation, you can consider this. The other thing is that I absolutely love what you showed with this reduction of disulfide bonds, that you have this reverse spooling, as you say, yeah? And that you suggest a beautiful model that this flagella, in fact, movement and rotation is associated with morphology and with the compactness of the sperm head. These are very interesting for infertility things in the wider context. Very interesting, because morphology and sperm motility are very these are beautiful data also to know for andrologists that I wanted to comment. Thank you very much. Yeah, it is beautiful.
I don't know what to say except thank you. No, maybe I can
send you our article 20 years ago and several reviews. Yeah, I'm still involved in these studies. Yeah, so a little bit acknowledge.
Yeah, I can comment though that yeah. So in mouse and in other insects, of course the histone protamine transition is a period of great stress for DNA. So you can see breaks. It's definitely part of the picture, right? And I don't know at our kind of scale how much it can contribute to the biophysical.
It can contribute.
Chromatin, but
if
yeah,
you have breaks, you should have more extended DNA. It should behave like different polymer properties. You should
depending on the repair machine. So we need to characterize this.
You just check it.
Yeah. And yeah, and also for the so I think that what the result for the disulfide reduction, what it really is showing is that the entire architecture of the nucleus is held together by the protamines, right? And so maybe even so at this point, if you break down the structure of chromatin, even the membrane doesn't hold it in place, right? It's kind of spoofs like that. So I think it's interesting to think about it in that way, which is kind of the opposite to what happens before when we think that the flagellum is operating at torque force. It means that at this point, the chromatin is very easy to influence by external forces. And so there must be a switching, a tipping point where the chromatin is very impacted by the development of the cell. And at some point, it becomes frozen by the disulfide bonding. And at this point, the forces are no longer necessary, but the chromatin integrity is very dependent on the protamine. So yeah, all these questions we have in our minds. Thank you for your comments.
Thank you very much. And we have a question in the chat from Maria. She's asking that you mentioned the 20 fibers per nucleus. Do they correspond to the number of the chromosomes that crickets have?
Yeah, so no, sorry. So we have, when we look at the fibers, it's 200 of the fibers going up and down. Of course, there must be chromosomes going back and forth at least a few times. The precise size of the chromosomes is not known, so we cannot really actually estimate this. But yeah, we expect that chromosomes are actually bending and going back and forth at least a few times. Yeah.